Okay. Welcome, everybody. So we're going to start with the seminar, I think the last seminar of 2023. And it's my pleasure to have with us today David Richardson. We have tried a couple of times to get him here, and finally he is here, and we're really happy. So David Richardson is Professor of Evolutionary Biology and Conservation at the University of East Anglia in Norwich, at the UK. He works on a really large array of different things. We know him because of his work in evolutionary biology, but when checking carefully more his trajectory, he also works on behavioral ecology and conservation and touches a wide number of topics and works on a wide number of organisms. So the work that we know in my group we know best is his work on host-parasite interactions, the role of parasites in driving diversification evolution on host populations, and his work on immune gene evolution. But he also works on a large number of things, including senescence, cooperative breeding, genetics of sexual selection, inbreeding, and evolution of small populations. He also does work on experimental evolution as well. He works on different organisms, mostly birds. Seychelles warblers is what shows most often in his work. I guess not a bad place to work or not a bad system. In this system, he works on gut, microbiota, senescence, inbreeding. But he also works a bit closer to us in the Bachelor pivot with Bisbita in Madeira and the Canary Islands. There are some other people at the museum working on the Canary Islands as well, where he works on pathogens, but also evolution on islands. But he also works on beetles, where he does experimental evolution. So, well, he's going to talk a bit more on the topics that we were most interested, but I hope he will give an overview of all what he does. And he also collaborates with a large number of people in different parts within the UK, Oxford, Sheffield, but also within Europe, Groningen, and also collaborates with the Universidad de Oviedo. Mm-hmm. And so now the floor is yours.
Hello, hola. I guess I first really have to apologize that I'm awful at languages and I know no Spanish. This is partly because I'm awful at languages, but partly because I'm English, and then I married a Danish woman and I have to learn Danish, and that's already hard enough. So I apologize for that. Okay, yeah, so today I'm going to be talking a lot about host pathogen systems, and specifically about pathogen-mediated selection, and how I've spent quite a lot of time trying to understand pathogen-mediated selection. Okay, by the way, also, because I'm not speaking Spanish, if I'm speaking too fast or if you can't understand me, just tell me. So yeah, host pathogen selection. It's really important because it's a key driver of genetic variation, especially when we look at things like pathogen-mediated balancing selection. And because of this, it's really important that we understand how it functions in terms of evolution and enabling adaptive potential, the importance of it in conservation and enabling the fitness of individuals and the persistence of populations, and even understanding, whether it be birds or humans, understanding generalities about interactions between pathogens, and by pathogens I mean parasites and microbes, macroparasites and microbes, and health. So I think it's a very important topic overall. I'm giving no prizes for you guessing which islands those are. The key point here is, I think, understanding the impact of pathogen-mediated selection is particularly important nowadays. It's important when we look across natural islands that we understand how it interacts between different populations and the host. And that's particularly important because now the world is becoming more and more fragmented, and organisms are generally living in small, fragmented populations because of human impact. I'll just outline the talk structure. First of all, I'm going to be talking about the pathogen-mediated selection in the island birds that I work on. I'll talk about how we identify which parts of the genome interact with the pathogens. And then I'll show you a case study of how we follow up with those, say, candidate genes and look how they are impacted by pathogens and how they evolve within a system. And then I wanted to go on to more recent work that we've been doing that comes from that pathogen-mediated selection, but is looking at the causes and consequences of gut microbiome variation. So the overall point is to understand how pathogens stroke microbes shape host variation, because not all microbes are pathogens, of course. So I'm going to start by talking about the island bird systems that I work on. The first one, the Bispetta caminero. That's as much Spanish as you're going to get. And I started this with Juan Carlos Ilera, who works in Oviedo now, in 2005. But the work I'm going to be showing you today is mainly the work by Eleanor Shepherd, a PhD student. The other system I work on a lot is the Seychelles warbler, and I'm going to be talking mainly about the work that Charlie Davis, who's now a doctor, has been doing on that. But you can see generally working on boring little brown birds, but in very nice places. And we'll start by focusing on the pipit, the Bispetta. So, as I'm sure you know, the pipit lives across these islands. It came from mainland Africa and then expanded up. We know exactly when that did because we've got genomic information, and we've dated the colonization of Madeira to about, the colonization of the Canaries to about 200 million years ago, where it diverged into the Bertholet's pipit. Madeira about 50,000 years ago, and the Savagens just 8,500 years ago. Whoops. Now, the other thing that makes these islands a really good kind of system to look at host pathogen interactions is, as well as having all these different islands, we have different pathogens that live on these islands, and those pathogens differ in frequency on different islands, and I'll get back to that in a minute. We've also done a lot of work looking at some key genes across these populations, and we've been looking at signatures of past selection in the genome. And we found strong signatures of past selection in the MHC and in certain TLRs. And I'll just quickly introduce these. I know lots of people here work on things like the MHC, but both these sets of genes basically code for molecules that sit on the cell membrane and bind antigens from, pathogens, anything that actually gets into your body, and they recognize non-self antigens, and they create an immune response. The MHC does that for the acquired immune system. The toll-like receptors do that more for the innate immune system. So they detect pathogens and they trigger immune responses. They are incredibly polymorphic, some of the most polymorphic genes ever found. And when you look at alleles in these genes, they often are associated with specific pathogens, with resistance or resilience to specific pathogens. Now, in the pipit, we find actually that there's quite high levels of variation in some of these MHC genes and these toll-like receptors, despite the fact that the pipit went through these bottlenecks in colonizing the islands. And it seems that some kind of balancing selection, some mechanism has maintained variation at these specific loci. So, for example, in the MHC, it seems that after colonization of our islands, because the MHC is a set of duplicated genes, you get a kind of gene conversion that reshuffles the pack and makes a lot of genetic variation again, even if initially there's almost none there. I'm not going to be talking about that today. Today I'm going to be talking about specifically how we then look at key pathogens, and in this case it's the avian pox, and how we drill down into understanding whether those key genes, like the MHC and toll-like receptors, are playing a role or interacting with that pathogen. So a specific aim is variation at those key genes where we've identified signatures of selection. Is it associated with pox infection? So is that pathogen potentially creating this balancing selection? But also there's many other immune genes, obviously, in the genome, and we want to be able to identify other immune genes that may play a role in dealing with pox infection. So we're trying to do that as well. Now, I won't give much methods in this talk. We're just kind of having an overview. But obviously, we go out and we catch the birds, and we've been doing that for many years. We've actually caught in 2022 and 23 as well. And when we catch those birds, we obviously take a small blood sample, but we also screen them for pox infection. And it's an obvious phenotype that you can see. So we've done quite a few birds over the years on those different islands. We've also done screening of different TLRs, and we find this one TLR, TLR4, shows very distinctive patterns of the generation of variation across islands. So it seems like something important's happening at this TLR. We've also screened for MHC class 1 variation because we know that seems to interact with malaria, and we've got various papers on that based on screening at a landscape level across Tenerife. And then our first genomic approach was using RAD sequencing, and we sequenced 22 individuals from 13 different populations so that we've got genomic data. Now, the first thing we want to do is we want to look at, whether an individual has pox, and these are the signs of pox, and whether that phenotype, being infected or not being infected, is associated with the variation at MHC class 1 and TLR4 genes. And I don't want to bore you too much about this because basically we did lots of work and we found absolutely no association. This happens. It's a lot of work for someone to prove this, but we don't find any association. And that might be true. It might be just no association, but there are some problems with looking at variation in this way with individual-based infection. So if we're looking at individuals that we capture in the wild, we might have individuals that have pox, and we might say, okay, these individuals are susceptible, right? And that's what we term them as. But actually, we're only looking at the individuals that are probably resistant to a certain degree, because although they have pox, they're the survivors. The wet pox would kill off most birds, and only the survivors end up with these scars of the pox. So that data is a bit confounded. Equally, if you look at individuals and they don't have any sign of pox when you get them in the wild, it might be that they're totally resistant to getting that infection, or it might be that they've just never been exposed to that infection, right? So again, that leads to very confounded data. This is a problem on working on a wild system. So we wanted to think of ways to get around that and still identify which genes play a role with pox infection. And we realized that we'd got quite good population level data on pox. And remember, we've been doing this for 20 years. So these patterns of differential pox prevalence on different islands are consistent across the 20 years. They are actually also consistent with basic biogeographic rules. In general, bigger islands, islands closer to Africa, have more pox than smaller and more isolated islands. So we think that this will indicate, at least to some degree, that there's been consistent differential selection between these populations in terms of how much pox they've had. What we then want to do is use the genomic data that we've got from these different populations and do what's called a genotype environmental association analysis. And what we're doing here is we're looking at the allele frequency at all those SNPs across the genome, and we're linking it to environmental variation. And in this case, the environmental variation between the populations is the pathogen, the pox prevalence. And this model also controls for the neutral population structure, so it controls for that. So when we do that, what you derive from this method is you get a Bayes factor here, but you can also use an absolute Spearman's rank correlation just to rule out outliers. And basically, you take any SNP in your genome that is higher than the Bayes factor threshold of 1% and the Spearman factor threshold. If you do that and you look at many different SNPs here, because we're just using the RAD sequencing data, we don't have tens of thousands of SNPs, but we have quite a few. But remember, the Berthollet's pipet is also has low genomic variation. The key thing is we've got some genes that stand out here associated with pox prevalence in those islands after controlling for neutral population structure. And that gives us, they're linked to 12 candidate genes, and so we can identify what those genes are. And I'll give you an example of just one. So this one of the best ones, is linked to a heat shock protein, and we look at that, and then we say, okay, what information do we have about that gene? Well, that gene's actually already been found in humans and in mice to be involved in the replication of pox, or stopping the replication of pox virus. This is just one example, but I can go for all 12 of those, and they end up being a very logical set of candidates because all of them are involved in like stopping the replication of viruses, etc. It's really worked a lot better than I expected it to, actually. They seem very logical, but they are only candidates. We haven't proven yet in the Berthollet's pipet that they are actually interacting directly with the pox virus. So just to summarize, we've got no associations between TLR4 or MHC class 1, despite the fact that there's these funny signatures across the islands with that. But we do find these associations with pox prevalence at these 12 loci, and that provides us with these candidate genes that we can focus on and look at in much more depth. And they're a very logical set of candidate genes. So that's very reassuring, I guess. Next steps on the pipet, because we haven't got further than that, is we're actually going to screen pipets and look at, because we've got now 60 genomes, full genomes from contemporary populations, we want to screen genomes from museum samples taken about 150 years ago. You're in a museum, right? You know this type of thing. When I worked on other stuff, I went around various museums and they said, oh, we've got 2 Seychelles warblers. You can have a little toe from 2 Seychelles warblers. When I went to the Natural History Museum in London and said, do you have any pipets, Berthollet's pipet? They pulled out drawers and showed me 500. Someone had gone to the Canary Islands with a machine gun for these guys. Not quite sure why they needed so many. But we have actually really excellent museum samples. And now we're doing, we've screened 58 museum samples, full genome sequencing. So we're going to look at more depth at how certain genes, immune genes, have changed over time. What's happened to our candidate genes over time? So that's as far as we've got with that on the pipet. But I wanted to then go on and show you what we would then do with these candidate genes potentially. And I'm giving you an example from what we've looked at in the Seychelles warbler, another little brown bird. And this is linked to TLR3 in this case. So here's the Seychelles, obviously where Seychelles warbler comes from. This is the island that I work on there. it was the last place where Seychelles warblers were in the world. It was down to just 29 individuals left in the world on this one island. And that's obviously been preserved. You can walk around this island in about 15 minutes. It's small, but there's about 300 adult birds on the island. Just to give you an idea, even to get them to colonize this island, we had to catch them, put them in a boat, and drive them over there. They will not fly over water. It means that we can watch every single individual in that population. We've got them all color-ringed and bled, and we can follow everything they do, how many offspring they have, who they mate with, everything, basically. When they die, we know exactly when they die. Well, exactly, within a few months. So it's been through this bottleneck, the Seychelles warbler, so it has really low genomic variation overall. But if you look at immune genes, there's higher variation in the immune genes. Certain immune genes seem to be maintained through balancing selection. And we can look and see particular signatures in the DNA that suggest that or confirm that. And we've done a lot of work looking at the MHC and looking at things like female mate choice and the MHC. Won't go into that today. And we've also identified signatures at one key immune gene, TLR3. But when you look at signatures in the DNA, you are looking at past selection or past processes, not necessarily contemporary processes. So it is possible that you're just looking at the ghost of selection past. So we want to now find, are those genes that show these signatures of selection, are they actually evolving now because of selection? And I'm going to focus on that TLR3. It's a viral sensing receptor. We find this SNP in a key binding site of the receptor, and it has quite high minor allele frequency, this SNP. So what we're going to do is we're going to take lots of our individuals. We've basically got thousands of individuals that we've monitored ever since we've been working on the warbler, since about 1994, for me. And we're going to see if that TLR3 has been evolving in the population, and if so, what is driving it? Actually 1992, but I started actually in 97. So we've got blood samples from all these years. We've got many individuals, but I've just picked some cohorts and screened those. We've got over 1,000 individuals. We've also got some individuals from populations where, for conservation reasons, we translocated the birds. I won't go into the methods, but we can screen very effectively this SNP at this TLR3, the SNP that seems to be under selection, and we get these genotypes. You can get the AA, the AC, and the minor allele homozygote CC in those proportions overall. So that's, yeah, you can get more detail in the paper there. What we want to see then is variation at this allele still evolving? Is it still moving in our time frame? So what we do, looking at birds that we've caught over the years on Kuzan Island, this is Kuzan Island, the field site, and seeing if the frequency of that SNP changes. That is evolution. And this is a pattern we get. Over time, the frequency of this C allele is decreasing from 39% back in 93 to just 28% now. That's on the main island, the original island where these birds were kept or survived, but we've translocated them to all these different islands over the time. And so we can look at the frequency of this allele in the population when we translocated it and the frequency if the frequency changes. And that's the pattern we get. So we get consistently the same pattern of this SNP decreasing in frequency at this key TLR3. By the way, just for anyone interested in the conservation, across these islands now we have about 3,500 birds, and it's been taken down to just vulnerable on the red data list. So it's been a big success for conservation as well. So, yes, this gene is evolving. So what's driving its evolution? Well, first of all, we decided to look at survival. Is there an association with the haplotype? So you've got the minor allele, homozygous, the heterozygote, or the major allele, homozygous, and we can look at their survival. These birds live for quite a long time, but we've been following them for many, many years, so we can get that data. Because they can't even fly to other islands, we have very good data on when they die, basically. And that's the pattern that we get. We find that the CC genotype is having reduced survival, and that's equal in both sexes. A 37% decrease in survival if you're homozygous CC, no difference between the heterozygote and the major homozygote. And it generally seems to happen in that first year, which is not surprising. We know that's when most mortality is happening. Now, the other thing we looked at was lifetime reproductive success. Because we genotype every bird, we do DNA profiling on every individual, so we know their exact genetic reproduction across their lifetime. And this is the number of fledglings that recruit for an individual, and we can look at that. Now, if we look at it for females, you can see again that there is a decrease in the lifetime reproductive success of individuals with CC, but it's not significant. But if you look at males, they have a very significant decrease in both the heterozygote and the minor allele homozygote, showing that any individual that has a C allele has a lower reproductive success over time. So this is how we've been focusing in on exactly how this particular SNP that is causing a change in the binding site of a viral sensing receptor is influencing evolution in this system. So we've got strong TLR3-based selection, especially on males. So just conclusions on this bit, we see that there's this contemporary evolution at this particular gene. We know that natural selection, viability selection in young individuals plays a big role. I didn't want to go into it today because it's overly complicated and we're still working on it, but it does seem that in males, sexual selection plays a role as well. And yeah, we'll leave it at that because we haven't finished that work. But obviously that leaves you with some unresolved questions. First of all, this gene, the TLR3, seemed to be under balancing selection. So it seemed to be maintained in the population despite the fact that many other parts of the genome had almost no variation. However, we are showing that it's decreasing in frequency, right? So that's not evidence of balancing selection. That's evidence of positive selection against that allele. So what might have been maintaining this allele? Well, we've got no evidence of heterozygote advantage either in this system. But the other mechanisms of balancing selection are things like frequency-dependent selection and fluctuating selection. And they can happen over very long time frames. Now, we might just be seeing this decrease that's happening at the moment because it's part of a pattern of frequency-dependent selection that might be happening over absolute decades. So how can we look at that? Well, I can try to persuade the funding bodies that they have to give me 50 years more funding because if this pattern carries on happening, the C allele will go extinct in 50 years. Unfortunately, I don't know if I'll still be, well, I do know that I won't still be around at that point. So that's not a good idea, but what we can do instead is we can go back to museum samples. And even based on this, if you project it back, this allele would have been at equal frequencies only 20, 30 years ago. So it would be really interesting to see what the allele frequencies were when we look at museum samples going back to the 1880s. So that's where we are on this question with the Seychelles warbler. The other key question, obviously, that you might be thinking is, okay, so what is causing this difference? What is the selective agent? What is the virus? We don't know. We go on that island. We know the bird has got malaria, just one strain, GRW1. We can't find any other pathogens. We can't find, it's got no nematodes. It's got no coccidia in its gut. It doesn't seem to have any obvious symptoms of any disease. But, what are we looking for? How would you identify if, the warblers had COVID, right? Say they had COVID, what would they be doing? Well, if they were really poorly, they'd be hiding in a bush and we wouldn't catch them, or they'd die and we wouldn't catch them. Otherwise, we're looking for birds that are going around coughing, which is not that easy. So what we've got to do is find a different way to look for the selective agent. So the TLR3, it binds double-stranded RNA. So we're taking RNA samples for the last couple of years now, because it's probably an RNA virus. And we're looking hard to see if we can generate the virome of the Seychelles warbler, all the viruses that exist in it, and whether we can identify associations between key parts, especially RNA viruses, and that TLR3. So that's where we are at the moment with that. Okay, so the final thing, quickly, hopefully, got time, is looking at the gut microbiome variation. So I've said before, we find no large parasites in the gut, but of course, the gut microbiome could contain many things that are interacting, not necessarily as a pathogen, but could be good or bad and interacting with the host immune system. So we thought we'd have a look at the gut microbiome, partly for that, and partly because we've done a lot of work on senescence and variation in senescence within population, and we wanted to understand the gut microbiome's role in that. But I'm not going to go into that today, just about pathogens and the gut microbiome. So I'm sure, it's a big topic at the moment. The gut microbiome plays this complex community of microorganisms that exist in the gut of most vertebrates, plays a big role in all these different processes, right? And it can have good and it can have bad effects. You can have pathogenic microbes in there. You can have microbes that are sometimes good and then can be bad in certain situations, etc. And another key point is you often find high individual variation in the gut microbiome. So there's lots of factors that can interact and play a role in determining what's in the gut microbiome. Environment, diet, etc. But what we specifically want to look at is how it's interacting with the host's genome, right? So we're looking at here trying to resolve the environmental variation so we can see, for example, how much the immune system might play a role in shaping the gut microbiome and vice versa. So obviously, we know that the immune system plays a big role in determining what bacteria or other microbes are in the gut. It's a very complicated role because it has to maintain beneficial bacteria or beneficial microbes, but it has to also keep out negative or pathogenic bacteria. And in this light, again, it's that MHC, the major histocompatibility, that's come up as a key player. And there's quite a lot of work done in the lab with things like mice showing that there's interactions between MHC variants, and gut microbiome composition. However, there's two things about that. One, we know captivity could quite radically alter the microbiome. And two, if we really want to know what's happening in the wild, what lead to the evolution of different species, what changes the fitness of different individuals, we need to do more looking at whether this is a significant relationship in the wild. And so again, we go to the Seychelles, where we can do this on this island. Our aim is to look at immunogenetic variation, the MHC. Does that shape the gut microbiome? And is that gut microbiome variation associated with fitness? To do that, we've been taking, since 2017, fecal samples. You'd never know how excited we get nowadays when we get a good poo sample. It's a freaky thing to be, oh, look at that. That's really impressive. But it means that we can get good sequencing of the variation. We've been using 16S for the bacteria, ITS2 for the fungi. We've also still been sequencing from the blood samples of the host, the warblers, for the MHC class 1 and class 2, and I won't go into the methods. But for the MHC, we find various alleles in the Seychelles warbler. This we've covered many a time, but here you can see we're focusing on these variable alleles. These are so rare that we can't do much with those. That's the MHC class 1. MHC class 2, we have a few fixed or almost fixed alleles and some very rare alleles. Again, we have to focus on these polymorphic ones. So that gives us our MHC screening. We've then been screening the gut microbiome. This graph here shows that we've got 244 different birds, but actually a lot of the birds are found in each of these classes as they get older. So we've got about 1,200 samples that we've run now. And that shows that you get what is kind of classically thought of as a very typical passerine bacterial gut microbiome. Protobacteria, firmicutes, actinobacteria. This is what you'd find in an insectivorous passerine in Europe as well. But it also shows that there's a lot of variation. Each of these lines is an individual, and the colors show these different phyla of the different bacteria in this case. So we've got a lot of individual variation there. Now, we can look, when we're looking at the relationship between the MHC and the gut microbiome, we need different metrics. For the MHC variation, we look at the MHC diversity overall, so how diverse an individual is at this suite of replicated genes. But we also look at the presence and absence of key MHC alleles, specific MHC alleles. We also have two metrics of the gut microbiome variation. We can look at the alpha diversity, just a number of different ASVs, species, whatever you want to call it, microbes that exist there. That's an individual level thing. And we can also look at the beta diversity, the difference in the gut composition between different individuals. So when we look at the association between the MHC variation and gut microbiome variation, what do we find? Well, MHC diversity has no effect on gut microbiome diversity or composition. However, if we look at specific alleles in the MHC, they really do seem to change the diversity and the composition. And I'll give you more detail on that. So here's the main alleles that we've been looking at. Here's the effect size of those alleles on the gut microbiome. Positive effect, increased gut microbiome diversity. Negative effect, decreased gut microbiome diversity. And you can see that we've got these key alleles. Do you want me to press? Oh, come on, I'm back. Yeah. That have a very significant effect on the gut microbiome diversity. We then looked at specific alleles, and if we look at like key alleles here, those that are having an effect on the composition, this is complicated, but it's looking at the different allele. Each box is a different MHC allele, and the points are the relative abundance of types of bacteria in this case, with or without that allele, right? So this side is without, that side is with. And you can see that these certain alleles are very much associated with an increase in the abundance of certain bacteria, Lactobacilliaris here, which is generally thought of as a positive type of bacteria in the gut of many birds. But also you can see it's related to variation in the presence of this I can never pronounce this right, Cornea bacterialis, a mycobacterium strain, which is generally seen as a pathogenic thing. I think it is involved in what they call, is it avian pseudotuberculosis or something like that? It's generally seen as a pathogen. So does the immunogenetic variation shape the gut microbiome? We've got papers on the bacteria and on the fungi where it both seems to have an effect and independent effects. The question now is, does that gut microbiome variation, the change caused by those alleles, affect host fitness? And again, we can look at all those birds that we've had over the years and see how long they survive. But because we've only been taking the gut microbiome since 2017, we can't follow them throughout their entire lives at this point. So what we did here was just looked whether or not, when we sample a bird, does it survive to the next season that we're out there, which is normally about 5 months later. So does that bird survive in the short term? If we look at the gut microbiome diversity, we find no difference in who survives and who died over that short term with gut microbiome diversity. But if we look at the composition, there is a small but significant difference in those that died and those that survived, suggesting that individuals that die have slightly different gut microbiome. And if we look at that in detail and look what specific bacteria are in those that died on this side versus those more in those that lived, you can see there's a load of different bacteria that are more prevalent in individuals that were going to go on and die within 5 months. And that Cornea bacterialis comes out again. That's this Mycobacterium avium, and that's the one that was increased or decreased in frequency by the MHC alleles. And it's that one that we're going to be following up now to really investigate that in more detail. But that seems to be playing a role in mortality in the warbler, and it seems to be linked to the MHC, the immune genes of the warbler as well. So just to summarize this work, so we have different MHC alleles associated with gut microbiome diversity. Gut microbiome diversity is not associated with survival overall. Why do I keep saying this? Well, I keep saying this because in general, in mice and in humans where it's been done, people have identified quite a close association between alpha diversity at the gut microbiome and survival. However, the general thinking is a lot of that change in diversity might actually be due to behavioral traits. If you get old, you start having to take lots of medicine. I know. I've been having to take lots of medicine for a bad back. You may change your diet. You may change your social life. You may end up living in a care home, and that will radically change your alpha diversity. So whether or not it's really associated with survival in humans is still a debatable point. But gut microbiome composition is affecting survival in the Seychelles warbler, and it's a specific microbial ASCs linked to survival. So this is really interesting because there seems to be this link between the genome of the host, the bird, between the gut microbiome within the host and survival. So it might be that there's this whole complex pathogen-mediated coevolution that's maintaining immunogenetic variation in the warbler. So hopefully, coming to the end, I've explained how we're looking at host-pathogen interactions across fragmented island populations, how we can build that up and look at the interaction, and how we're using things like genomics, etc., to investigate that in more detail. And with that, I'd just like to thank everyone who's worked on these different systems. A lot of people, a lot more than even here, but I put the main players in there, and ask you if you've got any questions. Thank you.
Okay, thank you very much, David, for this really interesting talk. So do we have any questions for David?
Thanks for the interesting talk. I wonder if in the second study you show, it is possible that the pathogen disappears and the genes are still there, decreasing every year?
In the Seychelles warbler, the TLR3, yes, it is possible. And I guess that would be linked to kind of this idea of fluctuating selection, which can, if for 20 years a pathogen is there, it might have selected for the C allele. then the pathogen disappears, and there might be a cost to that C allele. And in fact, we often find that there are costs associated with key alleles involved in resistance in the immune system. So it could be that it's just drifting down now because that pathogen isn't there. Now, I guess over the long term, it'd be really interesting to look at what the frequency was in the past. Is that allele going to just die out? Is the pathogen going to come back? I mean, it's really interesting to know, because at the moment, if that allele does die out, then that bird species, the warbler, will be more vulnerable in the future. But yeah, it totally could be that fluctuating selection and stuff. So it's actually something we think about quite a lot when we translocate the birds as well, because when we translocated the birds, we know they've got a strain of malaria. And when we were first translocating them, the vet said, you need to only take birds that don't have malaria, because you don't want malaria on this new population. And we actually argued, well, that's wrong for two reasons. One, we're trying to conserve biodiversity, and the pathogen is biodiversity as well. Two, we want the coevolution of that bird on a different island with the malaria to carry on, so that it doesn't then evolve without any type of malaria and a new type of malaria come in later. So it is really important that they keep, it's ironic, isn't it? It's important that they keep the pathogens to keep their immune health up. Good question.
Any other question?
Thank you very much for this very interesting talk. And I was wondering if you have done any work on post-transcriptional changes, and if there's any expression levels attached to these differences in allele frequencies.
We have, with the MHC, originally when we were doing the MHC on the Seychelles warbler with Helena Westerdahl in Lund, we did expression to show that those alleles were expressed. We haven't done anything with like the TLR or anything yet, but we should. And it is a deficit, I know. But we are actually, now that we're taking RNA samples, both for the virome and other things, we've got no excuse, really. Money might be an excuse, but yeah.
Any more questions?
It's more like a comment. Well, thank you so much. It was really nice. In your first project, you show the prevalence of pox in the Canary Islands, and you mentioned that maybe it's related to the size of the island, I think. And I was surprised because Porto Santo is a really small island and has a high prevalence. And I was wondering if you thought that could be possible, it could be related to the climate, because these Porto Santo, Lanzarote, and Fuerteventura are really arid islands. Yeah. Gran Canaria too, and I have no idea if this is related or something like that.
You're spot on there. The Porto Santo is a very strange population, because Desertas and Madeira, Maine have got nothing, right? And it's always the same when we go back. It's always the same. Porto Santo has more virus, so we can't. It does look like over time that there is an association with the drier islands, which is funny because actually you often associate pox being transmitted through water sources. But actually, if you think about it, on these dry islands, you often go to somewhere like Lanzarote and people, there's this tiny little pool of water that someone puts out and all the birds go there. And so they're probably all contaminating each other directly in that small water source. So it could be that there's an interaction with that. We haven't followed that up. We did actually have people on Lanzarote trying to link environmental factors with pox prevalence, and we started that in 2020, and we stopped it in 2020 for obvious reasons, basically. They had to all leave, and we never got back to that. So, but yeah, it's really interesting why Porto Santo is like that. Also suspicious it might be due to human factors, things like chicken farms and stuff. When we've done this on malaria, and we've done a lot of work on malaria as well, the main things that predict malaria in the pipit are water bodies, not surprising, and chicken farms. And we looked at that, and it's not actually that the malaria strain's in chickens. It's not that at all. It's just because of the food and the feces and everything else flying around chicken farms, you get a really high density of birds, and then you get a high density of vectors and stuff and transmission. So, yeah, it would be good to follow that up.
I have a question related to that. So even if you find the same patterns of infection over the years, do you still find differences? So is that changing even if the patterns are staying?
Yeah, there is variation in there. It doesn't explain all the variation at all, and it does fluctuate, but the overall pattern is generally consistent. Luckily enough, it means I have to keep going out and checking. So I've got my holidays sorted for the next 20 years there as well. And we are actually going out in March next year. We've got someone now looking at virome, how the virome colonizes with the pipit. So I can get to look again, but it does fluctuate, and we don't totally know why, but probably climate, probably other factors, but there is a core trend there.
I have a question about how you detect prevalence of pox. So you basically look at them and check for lesions. Is there a more fine-grained way of looking at it? So also, are some birds maybe asymptomatic and they have been infected and without producing these lesions? So that, if you don't find them, you don't just assume they are not infected, but they may have be asymptomatic but still be infected, and that may give you a final resolution?
We've tried to use PCR methods to amplify it. What we generally find, there was actually a study from people in Guyana about this, and they said that this method worked great for doing the blood, and 3 years later they retracted the paper because one technician had been making the data up. We chased that down for many years before, and we can't identify stuff from the blood, but if we get a fresh growth, it has to be a fresh growth, and we take a small sample from that, we can identify the virus. But if we take the blood from that same individual, we don't identify the virus using the PCR. So it's actually found it really hard to screen individuals for that. I guess we could go for some kind of antibody test to see if that bird has had it in the past and got rid of it. And what we normally find, though, even with those growths, is most of the growths we see are probably scars of pox that has been. From most of the growths, we can't detect viral DNA. It's gone, the bird has recovered, but it has these lesions, right? It's only when you get really fresh ones. So it would be great. The antibody one would help, because then at least we can rule out whether or not it's been exposed or not. But it's, again, complicated. That's a good question.
Do we have any additional question?
Normally, at least one person asks how they can get to go to that Seychelles island.
I have a question. Is the avian flu spreading over the world and causes very high mortality rates in birds? And I was wondering how this might affect your systems of study now and in the future.
We are starting to screen, obviously, with the virome stuff for that. The highly pathogenic strains of avian flu, the ones that are killing off all the seabirds, as far as I know, they don't get into the passerine yet. There are other viruses like Usutu, which is a form of West Nile virus, which is now in the blackbirds in Europe, and Sinbid is another one. So there are other things, and I think this is why it's important that we start screening the virome, because the few studies that have gone out there have looked at like a healthy population, say, of ducks, and they find tons of viruses are in there. And especially the warblers live in that island, the small island, contains about 300 Seychelles warblers, but it also contains over 1 million seabirds. It's absolutely covered in seabirds. It's an amazing island to go to. Just take a hat, because you're also covered in, yeah. And I'd be really surprised if there isn't transmission there. It's just that the highly pathogenic form of the flu hasn't got there yet. So I think this is why it's really interesting to start looking at virome, actually.
Just for curiosity, so you said you're going to test museum samples of the Seychelles warble for the TLR3. Do they still preserve the malaria strains, or can you also look at the pathogens of these museum samples?
I've thought about that. They will only give us the toe pad. And people I've asked can't get pathogens from the toe pad. I mean, I guess they don't even keep things like the liver in those stuffed birds, do they? So, I mean, obviously malaria embeds in the liver and stuff. So if you could get the liver, you could do it. But they've been stuffed. There's nothing. So we did think that because we're doing like whole genome sequencing from those toe pads, that we do have a kind of virome pipeline now set up so that we don't expect to identify many, but it should peel off any sequences that are from various different pathogens, including viruses. So, but I doubt we'll get anything from those. Someone was shaking their head saying, you don't get it from the toe pad, or did anyone know? No? Okay. I thought someone might be more expert on this than I, because I haven't tried it yet, but just people tell me it's not really possible.
Okay, so if there is no further question, we're going to finish it here, and we're going to continue in the kiosco. Thank you again, David, for this. Very nice.
Thank you very much for inviting me, guys.